So we will get started. Hi, everyone, and welcome back to another episode of Coffee with Infection Control. My name is Dr. Andrew Hurlbert, and I'm one of the infection control physicians here at Vancouver Coastal. I've been away for the past couple of weeks helping out in the interior. And before we get started, I just want to let everybody know that we will be taking a break next week while some people go on holiday. We will be back in the first week of August, again on Wednesday, and again at 2:30 in the afternoon, so you can pencil it into your calendars. We may be changing things to once every 2 weeks until the end of the summers, but we will keep you posted on the, I guess it's August 5th will be the next time we're here. So with all that, I will get started. For those of you that have watched this webinar before, you may notice that we have covered this topic previously. I am going over it again for two reasons. One is that unfortunately we had some foibles with our recording technology and we weren't able to record it and put it up onto YouTube to watch in the future. And the second is there have been a couple of updates, particularly around the more recent research around vaccines that I thought I would touch on, and we can go into a little more detail. As always, let me click on the right thing here. As always, to optimize your viewing experience for the remainder of this half hour, I would ask that you please turn off your camera feeds and mute your microphones. If, for whatever reason, you cannot hear or you are having difficulties with your video feed, please unmute and speak up, because there is no point in me continuing if people can't hear me and can't see me. As always, we have our Menti program running simultaneously. The Menti code for today is 260151. And if you would kindly join us on Menti, you'll have an opportunity to ask questions at the end of the presentation and to provide any feedback to future sessions and things that you liked and maybe didn't like. I just like to throw jokes in here every once in a while. So we are going to readdress the topic of COVID immunity. And to start, we're going to overview again the immune system and its basic components to give people a rough idea of how it works. We'll go over how the immune system responds to infection with COVID-19 and an update on where we are with our vaccines. If we have a little time at the end, we will discuss maybe some treatments and answer some questions. So, when it comes to the immune system, there are essentially four broad components. The first is a variety of barriers that prevent infectious or potentially infectious agents from getting into our bodies in the first place. These include our skin, our mucosal surfaces, such as the lining of our mouths, as well as secretions like our saliva, which contain enzymes and antimicrobial peptides, and other various secretions that can help fend off potential infections. There are several intracellular defenses that are very old and primordial, and these are systems that are in almost every single one of our cells that allow them to try and fight off any infection with a virus on their own without coordinating with anything else. Lastly, a barrier that is often overlooked is our natural flora, or the thin veneer of bacteria that we have on our external surfaces and lining our gut. This not only can work in coordination with our cells and provide us special nutrients and vitamins that we can't make on our own, but they actually act to prevent any bad bacteria, quote unquote, from getting hold and causing a problem. And so they basically occupy the potential real estate. The second broad category is the innate immune system, and this is our cave person immune system. I say that figuratively and not literally. Our cave people ancestors had much more than just this, but the general sense is that this is an immune system that can respond across the board. It does not necessarily need to have seen the pathogen or potential infectious agent before to result in some response that is effective in clearing viruses, bacteria, parasites, etc. It is composed of something called the complement system, which is a series of enzymes which can act to destroy bacteria in particular. There are several cells that participate and cause what we think of as traditional inflammation. These include things like neutrophils, macrophages, eosinophils, and then there are some cells that link the innate immune system to the adaptive immune system, which we'll talk about in a second. When I speak of the innate immune system, it's hard to do that without discussing the various mediators of the immune system, which allow the various cell types involved to communicate. These include cytokines, which are chemicals that you can see in the picture below with all the different text. You can see IL-3, IL-4, IL-10, TGF-beta, etc. These are messenger chemicals that allow cells to talk to each other over distance, and they allow the body to recruit cells to certain areas. Receptors are the way that cells can communicate with each other up close, or the way that they can take in their cytokines and translate them into a potential action. Antigen presentation is the complex process by which the immune cells can take elements of either bacteria or viruses or other potential infectious agents, digest them and convert them into a standardized way of displaying parts of the potential problem to the adaptive immune system, which allows the adaptive immune system to learn. Lastly, antibodies are things that most people are familiar with. These are proteins that are very specific to a number of potentially infectious particles and act to both inactivate them and to enhance the consumption and degradation of the little bad germs that might be floating around. When I talk about antibodies, there are several types, and they are all really targeted and characterized to their particular function. So, for example, IgM is the first antibody made. It is has the lowest affinity, so it has the lowest ability to bind on to bacteria or viruses tightly, but it compensates with that by having a whole bunch of different sites. So it's very big, and it basically allows the body to compensate for quality of binding with quantity. IgG on the far left is what we develop usually with a more mature immune response, and it is a much tighter binding, smaller molecule. The last major arm of the immune system we should talk about is the adaptive immune system, and this is composed of lymphoid organs, things like lymph nodes and your spleen, and it is associated with two major categories of cells. These are the T cells or T lymphocytes and the B cells or B lymphocytes. The T cells have a number of different subcategories, and their major role is in fighting off viral infections or infections that get within cells. The CD4 T cells also act as generals to coordinate the overall immune response. B cells, on the other hand, are best known for their ability to create antibodies and to create very specific antibodies of all types. And you can see here we've got a couple of diagrams showing the ability of these cells to kind of differentiate from a baseline naive B cell or T cell and change into very specific cells that have very specific functions and which are targeted to certain types of infection. In both cases, This is where we get our immune memory from. So both T cells and B cells have the ability to create memory cells, which can last for years and sometimes even decades and give us our immune memory. These are the key cells when we talk about vaccines and developing a durable and lasting immune response that allow us to see something at one point through a vaccine or through an infection, and then to be protected against it years down the road. Next, I have a couple of charts just to give a basic overview of how everything works all together. In this figure, you can see that when people first encounter a potential pathogen or microorganism, there is your first response is with your innate immune system. This is the response that will act in all cases across the board. It doesn't need to be specific, and that is what really helps to keep things under wraps initially. In the middle section here is where the innate immune system starts to talk with the adaptive immune system, and the adaptive immune system starts to learn and develop into these highly targeted cells or effector cells that can fight whatever the pathogen is very efficiently and effectively. In the final phase here, after you've cleared out your infection, you have the memory response with the green arrow, and this is what provides you that long-lasting memory. Now, when we look at the importance of the different arms of the immune system, they all have their potential pros and cons, and we really wouldn't survive without them. And by them, I mean without any of them. So our barriers are essential. If we think about people who unfortunately suffer large area burns and lose their skin barrier, they are very, very prone to potential infection. And it is a major concern, not only from bacteria around, but from even things that might float through the air and land on the exposed subcutaneous tissue. The red line in this diagram shows what occurs when you lack innate immunity. And so this is essentially a schematic looking at a summary of studies of mice and humans who don't have, for various genetic reasons, an effective innate immune response. Essentially, the infection goes out of control and takes over, and people die very quickly, or mice in this case die very quickly. The green line shows what happens when people or mice have deficiencies in their adaptive immune response. So they're able to control things a little bit better, as you can see, through their innate response. However, over time, the innate response proves to be ineffective in resolving the infection, and you can see that that green line continues to go up. The yellow line shows what happens when you have both an innate and an adaptive immune response which function normally. So I touched on this a little bit before, but here are a couple pictures of what how we think of our T cell and B cell responses, with our T cells attacking our own cells that might be infected with the virus and killing them off in a very programmed way, and our B cells, which produce the antibodies which neutralize various pathogens which might be floating around in our plasma or interstitial fluid. When it comes to immune memory, we see in the figure on the left what happens when we get that response to antigen A, which we'll call infection A. So just as we saw a couple of slides ago, there is a lag during which the innate immune system is working to control the infection and teach the adaptive immune response, after which we start to see a rise in antibody levels in response to infection A. And then over time, as infection A is cleared, those antibody levels drop back down. Now, when you introduce antigen A again, after the body has been trained, in this case where you have that antigens A and B, there is now a rapid secondary response, which produces antibodies right away. And this is a product of our memory response. This is a good example of our ability to learn from our first antigen exposure and then respond quickly and effectively when we see it again. The figure on the right shows that over time, without future exposure, our memory response does wane. So it doesn't just build up and stay the same. Over time, and this is time in years, we start to see our T cell responses and our B cell responses, or our memory responses, deteriorate and drop off. Now, what happens in COVID-19? Here we have a few figures that I'm going to walk you through. There are a number of different components on here which are excessively detailed for the purpose of this talk. So the general gist is that the virus is the circular icon here with the various spike proteins, which come into the cell, replicate within the cell, cause the cell to die through what's called either pyroptosis or necrosis or apoptosis, but in this case, pyroptosis. And when they replicate, they produce a bunch of new virus, which is then secreted back out into either the airway or the blood. And in response to this, the infected cell sends off a number of different chemical mediators or cytokines, as well as little bits of the virus, which are picked up by our immune system, our innate immune system, and which triggers the immune response. So initially, after the cell is infected, we can see that there are a number of cytokines which are released and which act to recruit other parts of the innate immune response, which will help to control things early on. And it will recruit elements of the adaptive immune response, which will then interact with the innate immune cells to learn. Now, there are two potential outcomes when it comes to COVID-19. What we see on the left is really a depiction of the worst case scenario, and that is a dysfunctional immune response. And this can happen for a number of reasons. Some of them are genetic, some of them are to do with an overwhelming infection, some of them are to do with concurrent medication use. But what happens is the innate immune response recruits the adaptive immune response, and it is inadequate to clear out the virus. So the virus continues to propagate, and in the meantime, you get more and more inflammation inside the lungs, to the point where people have difficulties breathing, require extra oxygen, and in some cases require intubation, ventilation, and in spite of that, still die. The figure on the right is what happens in a normally functioning immune system with a healthy immune response, and that is that the coordination between the innate and the adaptive immune system helps to clear out the virus and results in long-lasting immunity. So, the big question in all of this is, are people really capable of developing immunity to COVID-19? And the answer is yes. And we can see that because the majority of people get better. And in order to get better, you need to clear out the virus. So the very fact that we have the vast majority of people recovering from COVID-19 shows that human beings are capable of mounting an effective immune response to the virus SARS-CoV-2. Now, when we ask, okay, the next question is, really, how durable is that response? In other words, is that all due to the innate immune system? Do we have an adaptive immune system? Will that adaptive immune system last, and for how long? We can say, well, yeah, we have pretty good evidence to suggest that the adaptive immune system is triggered and develops at least a reasonably or months-long immunity. And we can tell that because we have detection of neutralizing antibodies in patients who have had COVID-19. And these are antibodies that will bind to the outside of the virus and protect the host from the virus getting back into the cells. And we still don't have any documented cases of people having had this virus twice. There are a couple of cases, particularly from South Korea, where they swabbed people again after they had swabbed negative, and these later swabs came up positive. But these were thought not to represent a repeat infection because those individuals did not have any symptoms and did not spread it to anyone else. They also weren't able to culture any live virus from these people, suggestive of either grumbling, burned out infection or maybe even some residual genetic material. So we know through this that, yes, okay, we can develop an adaptive immune response and that it lasts at least for several months. The pandemic has been going on since December, and we still don't have any cases of repeat infection. So we can say that the immunity developed from people who've had the infection likely lasts for at least 6 months. However, there are still some big questions. That right? Okay, good. And I think the big thing I want to point out is that antibodies really aren't the whole story. There are some very important things that can be drawn from the presence of antibodies, and that is, the presence of antibodies signal the development of an adaptive immune response, which is key. I think that that's what we touched on the last slide. We also see that the majority of people who have had COVID-19 recover without specific treatment, again showing that, we do have this adaptive response, which is great. And we know that these neutralizing antibodies are actually good in both the lab and in other people in protecting against COVID-19. So all of this is very good news. And lastly, which sort of touches on the last point of this presentation, we know that in the most recent vaccine trials, giving people these trial vaccines is capable of eliciting an antibody response, which again suggests they have an adaptive response, which is great. However, there are some pretty big questions which are still remaining. The first is that when we look at the people who have this dysregulated immune response and who unfortunately die from COVID-19, many of these people have very high titers of neutralizing antibody. And despite that, they still have ongoing detection of viral RNA. And so we know that the antibodies alone aren't doing the job in clearing out the infection. So if antibodies aren't sufficient, we need to know what else is required for potential immunity. We also know that, while this immunity lasts probably for at least 6 months, will this deteriorate over time? And can we correlate the presence or level of antibodies with our ongoing immunity? We don't know, and that will take time. We also know that as there are more people around the world who get SARS-CoV-2, there are going to be more mutations, and already there are several distinct subtypes of COVID-19. And while we don't have any clear evidence of this, it's not inconceivable that immunity to one version of this virus may not be conferred to other versions. So the last thing that is a little bit more worrisome is that there's some evidence out there to suggest that some people with mild COVID-19 infection don't actually mount a detectable antibody level. So in light of that, what does a positive antibody test really mean? When it comes to vaccines, these two questions I pose at the end have thankfully been answered to a degree. The first is that, following the vaccine, we are able to detect antibodies in certain participants. And thankfully, the vaccine designers are very smart people, and they concurrently check for the presence of T-cell responses and other immune responses to make sure that we have not just an antibody response, but an overall immune response. The last question is something that will only be found over time, and that is some viruses, there aren't that many, but viruses like dengue fever can produce this counterintuitive response wherein antibodies actually make the infection with other subtypes of dengue fever worse. And so there is some mild concern that if there continues to be differentiation in different strains of COVID, that antibodies to one strain could well. I think in light of not leaving people hanging, we'll go to some good news. And that is that last week there were two publications of the kind of preliminary results for two different vaccine trials. The first was in the Lancet from the Oxford group, which you may have seen a lot of news about. And the second was in the New England Journal of Medicine. And both of them looked at trial vaccines and whether they were able to elicit immune responses. And the summary is, this is very good preliminary news in that both these trials showed that the vaccine candidates were able to elicit both T cell and antibody responses specific to COVID-19. Both of them were safe for use in humans, and that the antibodies that were produced by both vaccines were able to completely neutralize the COVID-19 virus when they were checked in vitro, so in the lab setting. The Oxford Oxford vaccine candidate was furthermore tested against a COVID challenge in non-human primates. And what that means is they gave this vaccine candidate to a number of non-human primates and then exposed them to known concentrations at high doses of COVID-19. And they found that those primates which received the vaccine were protected against infection. Now, vaccine development is pretty tricky. And while this is good news, there are a number of steps that need to occur before we go from this very good news to having a vaccine readily available. And those include evaluations of long-term efficacy or durability, ensuring that the right number of doses are there to make sure that it's available everywhere, the logistics of making and transporting vaccines around to various countries, the prioritization of who gets it first, the logistics of storage and refrigeration, how we evaluate this in the long term, et cetera, et cetera, et cetera. So there are a number of things that are still required. However, this is not the first vaccine humanity has ever developed, so I think these things could be rolled out relatively quickly if we had a vaccine that successfully passed a phase 3 trial. So we've had a phase 1 and a phase 2 trial. We need to have a phase 3 trial, and that I think will be the trigger to start production. So what does this all mean? Well, the first thing that I hope you took away from this presentation is that our immune systems are very complicated. There are a lot of things that go into an immune response and multiple levels, and there's a lot of communication between the cells that are involved. The second thing that I hope you took away was that humans can develop COVID-19 immunity, and that this immunity is at least somewhat durable following infection. I also hope that there's an appreciation that the mechanisms of immunity are important and they have major potential consequences, and that antibodies really aren't the whole story. So there's been a lot of talk, or at least there was, especially about a month ago, about antibody tests and when those would be available and detection of antibodies, but really they are not the whole story, and there are lots of other aspects that we should be aware of. In a very, in a positive light, we have some very exciting preliminary data from a couple of good vaccine candidates, and I suspect that we will be hearing more and more about this in the weeks and months ahead. I think the most important part, though, is that we, at least in BC, have been very successful in containing the spread of COVID-19. And we can see this in the following diagrams. We can see the cases decreasing considerably, and this is in spite of opening up. So you can see on the top of this chart, you have phase 1, phase 2, and phase 3. These are our opening up phases. And you can see that our phase 1 control and phase 2 control were very successful. Our phase 3 has shown a bit of a rise. However, that seems to be coming down as well. When we look at hospitalizations, we see that they have gone down considerably, and critical care cases have also dropped to single digits. When we compare how we've done in BC with the rest of Canada and with the rest of the world, we can see that BC has actually done a pretty good job in limiting the number of new cases, number of deaths, and the number of overall cases. When we compare ourselves to other provinces, we are definitely better than average, and definitely better than average, especially given our population size comparatively. Most importantly, though, we have done all of this without any real effective treatments or vaccines. So what I take away from this, and what I hope you do too, is that even if we have a vaccine and or adequate treatments, the best and most effective, least costly, and easiest way to control or stop COVID-19 is with prevention and infection control. And I know I'm tooting my own horn on this, but I think that you don't need treatments, you don't need to worry about hospital capacity if you prevent people from getting the infection in the first place. So with that, I will leave you with our lovely website one more time, and I will bring it over to the Menti, which unfortunately anticipate we're going to have fewer questions due to our technical glitch, but feel free to log into Menti and enter any questions that you have, and I will gladly answer them to the best of my ability. Our code again this week is 260151. And we'll give everyone a couple of minutes. And I hope that while we are away the next week, we can sort out these technical glitches so they do not happen again. And I wonder if there is maybe another platform that we can be using. We'll do some research. If somebody could please post something on the question board for our Menti, that would be great, just to make sure that we know it's working. Again, test, perfect, thank you. Oh, we've got it. Well, does anyone have any other questions? We'll give it another 20 seconds. It doesn't look like it. Okay, well, if anyone has any feedback they'd like to give, I think we'll skip right to the last section. Please put down any feedback that you have, any topics that you'd like to see in the future, and if you have any last-ditch questions you'd like to have answered, please post them here under the what topic would you like to see at future sessions. Okay, all right. So you'd like to see no future topics, which is perfect because we're not doing a session next week. So we will see you all again in the first week of August. And until then, be kind, be calm, be safe, and try and enjoy some of the beautiful summer weather we're finally having. Take care.